Welcome, everyone. Thank you for being here today. I'm excited to introduce our presentation on hardwood anatomy. The title of our session is Wood 280 Hardwood Anatomy, and it's brought to you by AgPix. Throughout this presentation, we'll explore the structure of hardwoods, their characteristics, and what makes them unique. We'll cover the key concepts and details that will deepen your understanding of hardwood anatomy. Let's get started. Vessel elements are always present in the xylem tissue, and they play a crucial role in water conduction. They typically make up about 3 to 25% of the wood's weight and about 5 to 60% of its volume. Vessels themselves can vary quite a bit in size, ranging from just a few millimeters long to several meters, depending on the species and the part of the plant. Vessel elements are shorter and thinner-walled compared to other cells, with a large diameter that allows efficient water flow. Their length is usually between 0.3 to 1 millimeter, and their diameter ranges from 50 to 500 micrometers. One key structural feature is the perforation plates, which are openings at the end walls that facilitate water movement between vessel elements. The main function of vessels is the transport of water and mineral salts from the roots up to the leaves. To ensure smooth flow and prevent blockages, vessels have intervessel pitting, which are small pits that connect adjacent vessels. Some vessels also have spiral thickening in their walls, which provides strength and flexibility. Tyloses are another interesting feature. They are outgrowths that can block vessels, often during injury or aging, helping prevent the spread of pathogens. The porous nature of vessels can be classified into different types, such as ring-porous, diffuse-porous, and semi-ring-porous, based on how the pores are distributed along the growth rings, as described by Hoadley. Understanding these structural features highlights how vessels are designed for efficient water transport, which is central to the overall health and functioning of the plant. Today, we're looking at the red alder, scientifically known as Ulmus rubra. This species is quite important in our discussion of tree anatomy and wood structure. One key feature to note here is the scalariform perforation plate. This term refers to a specific type of vessel element structure in the xylem, which is crucial for water transport within the tree. The scalariform perforation plate is characterized by its ladder-like arrangement of elongated openings, which allows efficient flow of water. When I mention Hoadley here, he's a key researcher who described these features in detail, helping us understand how these structural differences influence the tree's function. Recognizing these structural elements helps us appreciate how red alder and similar species adapt to their environments, especially in terms of water conduction and overall growth. So, in the bigger picture, understanding these microscopic features gives us insight into the tree's physiology and its role in the ecosystem. Let's take a closer look at some of the structural features in plant vessels. On this slide, we see the simple perforation plate, which is a type of vessel end plate. It's called simple because it has just a single large opening that allows water to pass through easily. This kind of perforation plate is quite efficient for water conduction. Now, moving on to the scalariform perforation plates, which are a bit more complex. The term scalariform means ladder-like, and you can imagine these plates having multiple elongated openings arranged in a ladder pattern. This structure is characteristic of certain types of vessels and can influence how water moves within the plant. Both these types of perforation plates are key features when we're studying plant vascular tissue because they affect the flow of water and nutrients. Recognizing these differences helps us understand how different plants adapt their vascular systems for efficient transport. Overall, these structural variations in perforation plates are integral to the plant's ability to transport water effectively, which is a crucial part of the larger picture of plant physiology and adaptation. Let's take a closer look at intervessel pitting, as discussed by Hoadley. Essentially, this refers to the small openings or pits that occur between vessels within the plant's xylem tissue. These pits are crucial because they allow water to move from one vessel to another, facilitating efficient water transport throughout the plant. What's particularly interesting is that these pits are not just simple holes. They are specialized structures that help prevent the spread of air bubbles or embolisms, which can block water flow. So, They play a vital role in maintaining the plant's overall hydraulic stability. Understanding intervessel pitting connects directly to our broader theme of plant water transport mechanisms. It highlights how plants have evolved intricate structures to optimize water movement while protecting themselves from potential blockages. This balance is key to understanding plant resilience and efficiency in water use. Let's take a closer look at the connections between vessel elements in hardwood. One key feature we see here is the perforation plate. This structure is essential because it allows for efficient water flow from one vessel to another, facilitating the transport of nutrients throughout the plant. Next, we have intervessel pitting. These are small, specialized regions on the vessel walls that enable water to move between adjacent vessels without the need for perforation plates. They act like tiny gateways, ensuring smooth and controlled flow. Together, these features, perforation plates and intervessel pits, are crucial for understanding how water moves efficiently through hardwoods. They highlight the sophisticated adaptations plants have evolved to optimize water conduction, which ties directly into the overall theme of plant vascular systems and their complexity. So on this slide, we're looking at different types of vessel arrangements in hardwoods, specifically semi-ring-porous, diffuse-porous, and ring-porous woods. These terms refer to how the pores, or vessels, are distributed within the wood structure. Starting with semi-ring-porous woods, you'll notice that they have a pattern where larger vessels are concentrated in a zone that resembles a partial ring, but it's not as distinct as in ring-porous woods. This creates a somewhat transitional feature, and it's quite interesting because it shows a mix of characteristics. Moving on to diffuse-porous woods, these have vessels that are evenly distributed throughout the growth ring. This means the pore size and density are fairly consistent from the earlywood to the latewood, leading to a more uniform appearance and flow. Lastly, ring-porous woods, which Hoadley describes, feature large vessels concentrated mainly in the earlywood, forming a distinct ring. This pattern is quite prominent and is often used as a key identifying feature in wood identification. Connecting this to our overall theme, understanding these pore distribution patterns is crucial because they influence the wood's properties, such as strength, how it absorbs moisture, and even its appearance. Recognizing these differences helps us better utilize and identify various hardwoods in our projects. Now, let's take a closer look at pores and how they're described in cross-section. Pores are not just random openings. Their position relative to each other can tell us a lot about their function and structure. When we examine them in cross-section, we can see how they are arranged, whether they're clustered together or spaced apart. Understanding this spatial relationship is crucial because it influences how substances pass through the skin. For example, pores that are closely packed might work together to facilitate a higher rate of secretion or absorption. This detailed perspective helps us better grasp the complexity of skin anatomy and how it adapts to various functional needs. And of course, this understanding ties back into the overall theme of how skin structure relates to its function, something we'll explore further as we go along. Let's take a closer look at slide 9, which focuses on spiral thickening in vessel elements. Here, we see the concept of a simple perforation plate. The spiral thickening refers to the way the cell walls of vessel elements thicken in a spiral pattern. This structural feature is quite important because it provides strength and flexibility to the vessel, allowing it to withstand various stresses. The spiral design also plays a role in controlling the flow of water through the vessel, making it more efficient. When we talk about simple perforation plates, we're referring to the openings at the end of vessel elements that facilitate water movement between cells. The spiral thickening is often connected to these perforation plates, as it helps maintain the integrity of the vessel while allowing for efficient conduction. Overall, understanding these features helps us appreciate how plant vessels are specialized for optimal water transport, balancing strength and functionality in the plant's vascular system. Let's take a closer look at spiral thickening in vessel elements. This is a fascinating feature where the cell wall develops a spiral pattern of thickening. It's particularly interesting because it plays a crucial role in maintaining the structural integrity of the vessel while still allowing some flexibility. This type of thickening is often seen in certain types of plants, especially during specific growth stages or in response to environmental conditions. Recognizing spiral thickening helps us understand how plants adapt their vascular systems to withstand mechanical stresses. Overall, these structural adaptations are key to ensuring efficient water conduction and plant stability. Now, let's take a closer look at hardwood cell types, starting with the fact that they are always present in hardwoods. They are quite abundant, making up a significant portion of the wood's structure. In terms of quantity, they can constitute between 40 to 90% of the wood's weight and about 20 to 75% by volume. These cells are long and needle-like, which helps them perform their functions efficiently. Their walls are thickened and heavily lignified, providing the rigidity and strength needed for support. Typically, they measure between 0.5 to 1.5 millimeters in length, and their diameter ranges from about 20 to 30 micrometers. The primary function of these cells is to provide mechanical support to the tree. When we talk about fibers, we're referring to these very long, sturdy cells that help maintain the structure of the wood. Understanding these cell types is crucial because they play such a vital role in the overall strength and durability of hardwoods. Let's take a closer look at the structure of hardwood fibers. First, we have the cell wall, which is the sturdy outer layer that provides strength and support to the fiber. Inside the cell wall, there's the cell lumen, the hollow interior space that plays a role in nutrient transport and storage. Surrounding the cell wall is the compound middle lamella, which acts like a glue holding adjacent cells together and ensuring the overall integrity of the fiber tissue. Understanding these components helps us appreciate how hardwood fibers contribute to the overall durability and quality of wood, tying back to our theme of material properties and their impact on processing. Let's take a closer look at the fiber cell wall thickness variation between basswood and elm. As you can see here, the thickness of the cell walls can differ quite significantly between these two species. Basswood tends to have relatively thinner cell walls, which can influence its mechanical properties and how it responds to processing. On the other hand, elm generally has thicker cell walls, which can contribute to greater strength and durability. Understanding these variations is important because the cell wall thickness affects not only the wood's strength and stiffness, but also how it interacts with treatments and finishes. This variation ties into our overall discussion of wood properties and how species-specific characteristics can influence their applications. Recognizing these differences helps us better select and utilize wood based on its structural features. Let's take a closer look at fiber cell wall thickness variation, focusing on yellow poplar and black locust. As you can see, these two species exhibit different patterns in their cell wall thickness, which can have significant implications for their mechanical properties and utility in various applications. Yellow poplar generally has a thinner cell wall, making it lighter and perhaps more flexible, while black locust tends to have a thicker cell wall, which contributes to its strength and durability. Understanding these differences helps us appreciate how each species can be optimized for specific uses. This variation in cell wall thickness is an important factor in the overall performance of the wood and ties back to our broader discussion on wood properties and their practical implications. Let's talk about the longitudinal parenchyma. This tissue is always present in the wood, and it's composed of small, thin-walled cells. These cells typically measure between 50 to 150 micrometers in length and about 20 to 30 micrometers in diameter. One important thing to note is that these cells remain alive in the sapwood when they are functioning, which is crucial for their role in storage. Speaking of function, the main role of longitudinal parenchyma is storage, providing a reservoir for nutrients and other substances. These cells are connected by simple pits, which allow for the movement of fluids and nutrients between them. Now, regarding their arrangement, you'll find them in two main patterns: apotracheal, where they are located outside the tracheae, and paratracheal, where they are situated around the tracheae. These arrangements are important because they influence how the parenchyma interacts with other tissues, especially in the boundary zones. Overall, the longitudinal parenchyma is a vital component of the wood's anatomy, supporting storage and facilitating nutrient movement within the plant. Today, I want to focus on the concept of hardwood, specifically looking at the longitudinal parenchyma. As Wilson and White have described, the longitudinal parenchyma is an important feature within hardwood tissue. It consists of cells that run parallel to the length of the wood, and these cells play a crucial role in the storage and transport of nutrients and water within the plant. Understanding the structure and function of this tissue helps us appreciate how hardwoods support the plant's metabolic needs. It also connects to the overall theme of how different cell types contribute to the strength and functionality of wood. So, in essence, the longitudinal parenchyma is a key component that ensures the plant's health and vitality. Aquipodimos ver dos exemplos de maderas tropicals, empezando con Emburana sirensis, que es una especie muy valorada por su belleza y resistencia. La foto muestra la textura y el patrón que caracteriza a esta madera, que además tiene propiedades aromáticas interesantes. Luego, tenemos grubia sp. Otra madera que también es de zonas tropicals, conocida por su durabilidad y aplicaciones en ebenistería. En la imagen, también se observa la presencia de parenchyma longitudinal, un tipo de tejido vegetal que ayuda en el almacenamiento y transport de nutrientes dentro de la madera. Este parenchyma longitudinal es clave porque influye en la estructura y función de la madera, además de ser importante en su identificación. Estas características resultan como la anatomía de estas maderas refleja tanto su adaptación a su entorno como sus propiedades útiles para diferentes usos. Todo esto conecta con nuestro tema general, que es entender cómo la estructura de la madera influye en sus propiedades y aplicaciones. Let's take a closer look at the longitudinal parenchyma. This refers to the tissue that runs along the length of the structure, and it's essential for understanding how the tissue is organized. Within this, we have two key areas to focus on, the apotracheal and paratracheal regions. The apotracheal area is located away from the trachea, while the paratracheal region is situated right next to it. Recognizing these zones helps us understand the tissue arrangement and its functional implications. Additionally, it's important to note the boundaries that separate these regions. These boundaries are crucial because they define the transition zones and can influence how substances move or how the tissue responds to various stimuli. Connecting all these points, understanding the longitudinal parenchyma and its subdivisions provides us with a clearer picture of the internal structure. This knowledge is vital for grasping how the tissue functions within the larger system, which ties back to our overall theme of tissue organization and function. Let's take a closer look at this slide. Here, Hoadley emphasizes the importance of understanding the key concepts thoroughly, but in a way that feels natural and conversational. What this really means is that when we're discussing complex ideas, it's crucial to break them down into relatable, understandable parts. He highlights that the most important concepts should stand out. These are the core ideas that drive the overall message of our presentation. By doing so, we ensure our audience can grasp and remember the main points. Finally, Hoadley reminds us to connect these points back to the overall theme of the presentation. That way, everything stays cohesive, and our message becomes clearer and more impactful. This approach helps us communicate effectively and keeps our audience engaged throughout. So let's take a moment to really understand what Hoadley is emphasizing here. He highlights that, when we're working with content, especially in digital environments, it's crucial to think about how users engage with it. The key point is that content isn't just static information. It's part of a broader experience that needs to be accessible, meaningful, and adaptable. Hoadley also stresses the importance of designing content with the user in mind, making sure that it can be easily understood and navigated. This connects directly to our overall theme of creating effective digital experiences. By focusing on these principles, we're not just presenting information, we're facilitating interaction and understanding. So, the takeaway here is to keep the user at the center of our content strategies, ensuring that what we create truly resonates and supports their needs. So let's take a moment to really understand what Hoadley is emphasizing here. He points out that, when we're examining content, it's crucial to go beyond just surface-level details. We need to delve into the underlying structure and purpose of the content, which helps us grasp its full significance. This approach allows us to see how different pieces of information connect and how they contribute to the overall message or goal. It's about understanding the why and how, not just the what. By focusing on these aspects, we can develop a more nuanced perspective, which is essential for creating effective content strategies. Remember, connecting these points to the broader theme of our presentation helps reinforce the importance of thoughtful analysis and intentional design. In essence, Hoadley's insights remind us that thorough comprehension and thoughtful connection are key to meaningful content development. Let's take a closer look at the ray parenchyma, which is always present in the wood tissue. You'll notice that its height can vary quite a bit, from just a few cells high to many cells, reaching lengths of 50 micrometers up to several centimeters. Its main functions are transport and storage, making it essential for moving nutrients and storing reserves within the plant. In terms of structure, ray parenchyma cells are connected by simple pits between them, allowing for effective communication and transfer of substances. When they are adjacent to vessel elements, we find semi-bordered pits, which help regulate the flow between different cell types. Now, the shape of these cells can vary too. Some are procumbent, meaning they are blunt-ended cylinders that are radially elongated. These are always present in the ray. Others are upright, rectangular cells with a long axis oriented vertically. These can sometimes be found as well. The cells can be either homocellular, meaning all the cells are similar, or heterocellular, with different cell types present. As for their arrangement, the conformation can be uniseriate, with a single row of cells, multiseriate with multiple rows, or even aggregate formations, where groups of cells cluster together. In terms of form, you'll see procumbent cells, which are radially elongated and blunt-ended, and upright cells, which are rectangular and stand vertically. Recognizing these variations helps us understand the diversity and complexity of ray parenchyma in different species and how they contribute to the overall function of the wood tissue. Now, let's take a closer look at the hardwood rays in a transverse section view. Here, you can see how these rays appear in different types of wood. Starting with aspen, you'll notice that the rays are relatively narrow and quite evenly distributed. Moving on to ash, the rays become more prominent and wider, which is a characteristic feature of this species. And finally, in red oak, the rays are quite distinct and can be seen easily as narrow, elongated lines across the section. Understanding these differences is important because the rays play a key role in the wood's structure and how it interacts with moisture and other elements. Recognizing these patterns helps us identify the species and also gives us insights into the wood's durability and workability. This ties back into the overall theme of understanding wood anatomy and how it influences our use of different hardwoods in various applications. On this slide, we're looking at how tangential and radial surfaces in wood expose rays that are visible to the naked eye. These rays, which are part of the wood's cellular structure, create distinctive visual features. They often give the wood a unique aesthetic appeal, adding character and depth to the surface. When we see ray flecks or ray ends, those are the parts of the rays that become prominent. Ray flecks appear as small, contrasting spots or streaks, while ray ends are the tips of the rays that can be seen when the wood is cut in a certain way. These features are especially important for identifying species and understanding the visual qualities of the wood. Overall, these rays and their exposure play a significant role in how we perceive and appreciate different types of wood, tying into the larger theme of wood's natural beauty and structural features. So, when we're talking about a tangential surface, the way the ray is structured really depends on how many cells it contains across its width. If you imagine a ray made up of just a single vertical row of cells, so one cell wide, that's called a uniseriate ray. It's basically a single cell thick and runs along the surface. Now, if that ray is 2 cells wide, we call it a biseriate ray, meaning it has 2 cells across. And when it gets 3 or more cells wide, then it's considered multiseriate. This distinction is important because it helps us understand the complexity and diversity of rays on different surfaces. It also ties into the overall theme of how plant cell structures and arrangements reflect their functions and adaptations. Let's take a closer look at some important terms related to the orientation of plant structures. First, we have procumbent, which describes stems or branches that lie flat along the ground but do not root at the nodes. It's a common growth habit in plants that spread horizontally. Next, upright refers to stems or branches that grow vertically, standing straight up from the ground, which is typical for many trees and tall plants. Now, moving on to tangential section. This is a specific way of cutting a plant or a structure to observe its internal features. When we make a tangential section, we're slicing parallel to the surface, often to study the outer layers or the arrangement of tissues near the surface. These terms and techniques are fundamental because they help us understand plant morphology and anatomy better. Recognizing whether a stem is procumbent or upright and knowing how to prepare tangential sections allows us to analyze plant structure more effectively, tying into our overall theme of understanding plant form and function. Now, let's take a closer look at the radial section, which is crucial for understanding the structural differences in plant stems. Here, we see two main types, upright and procumbent. Upright stems grow vertically, allowing them to reach toward the light, while procumbent stems lie flat or spread along the ground. Recognizing these growth forms helps us understand how plants adapt to their environments. The radial section, as shown here, provides a cross-sectional view of the stem, and it's a valuable way to observe the internal arrangement of tissues. For example, in upright stems, you often find a more prominent central vascular system, supporting vertical growth. In procumbent stems, the tissue arrangement might differ, reflecting their horizontal or sprawling growth habit. This illustration, credited to Hoadley, emphasizes the importance of understanding these structural differences. By analyzing the radial section, we gain insights into how a plant's internal anatomy aligns with its growth form and ecological strategy. This understanding ties back to our overall theme of plant adaptation and structural diversity. Let's take a closer look at some key features of hardwood anatomy, starting with hardwood rays. These rays are essential because they facilitate horizontal transport of nutrients and store carbohydrates, playing a vital role in the tree's overall health. When examining ray ends, we notice variations that can tell us a lot about the wood's structure. Now, moving on to multiseriate ray ends, these are characterized by multiple rows of ray cells at the end of the ray, which can indicate certain species or growth conditions. In contrast, uniseriate ray ends consist of a single row of ray cells, providing a different structural pattern. Understanding these distinctions helps us identify and classify different hardwood species more accurately. They also connect back to the overall theme of wood anatomy, how microscopic structures influence the properties and uses of different woods. Recognizing these features allows us to appreciate the complexity and diversity within hardwoods, which is essential for applications ranging from woodworking to forestry. Let's take a closer look at this model of water flow in hardwoods, based on Sayu's work. As you can see, the diagram highlights the different pathways water takes through the vessel, fiber, and parenchyma cells. The sizes and textures of the arrows indicate the relative magnitude of these flows, so the thicker or more textured arrows represent larger volumes of water moving through those pathways. The key point here is that water movement isn't just a simple straight shot. It involves multiple routes, longitudinal flow through vessels and fibers, as well as flow through ray parenchyma cells. These pathways work together to facilitate efficient water transport from the pith, which is at the center of the tree, outward through the bark. Understanding these flow dynamics is crucial because they impact how trees manage water stress and nutrient distribution. By recognizing the roles of different cell types and pathways, we can better appreciate the complexity of hardwood physiology and how trees adapt to their environments. So on this slide, we're talking about these bubble-like or membranous materials that can actually fill or block the lumens of vessel elements. These are known as tyloses, and they form through a fascinating process. Essentially, they result from the protrusion of a pit membrane combined with the growth of protoplasm into the empty vessel lumen. This growth comes from living parenchyma cells, which can be either longitudinal or ray parenchyma. The key point here is that tyloses can effectively occlude or fill the vessel lumen, which has significant implications for the functionality of the vessel, especially in response to stress or injury. Understanding tyloses helps us grasp how plants manage internal blockages and how they may be involved in processes like disease resistance or the development of heartwood. In the context of our overall theme, these structures are crucial because they demonstrate how plants adapt physically at a cellular level, protecting themselves or altering their vascular system in response to environmental challenges. Let's talk about tylosis formation. Tyloses develop through the larger pits in the cell walls, specifically those that are about 8 to 10 micrometers in size. These pits are present during the formation of the heartwood in a tree. As the tree transitions from sapwood to heartwood, these tyloses begin to form within these larger pits. It's interesting to note that most hardwoods in temperate zones contain tyloses in varying amounts within their heartwood. This variability can influence how the wood behaves and its durability. Understanding tyloses is crucial because they play a significant role in the tree's defense mechanisms and in the overall properties of the wood. The process highlights how the tree adapts and changes as it matures, especially during heartwood formation. So, in the big picture, tyloses are a key part of how hardwoods develop their characteristics, impacting everything from their strength to their resistance to decay. Now, let's take a closer look at tyloses formation in white oak. As you can see on this slide, this process is crucial in understanding how white oak's durability and water resistance are enhanced. Tyloses are essentially outgrowths of parenchyma cells that develop into blockages within the vessel lumens. This formation effectively seals off the vessels, preventing the movement of water and pathogens. Moving on, we have the distinction between sapwood and heartwood. Sapwood is the outer, living part of the tree that conducts water and nutrients. It's typically lighter in color and more susceptible to decay. Heartwood, on the other hand, is the inner, non-living core that has become more resistant to decay over time. It often contains deposits of extractives that give it its darker hue and contribute to its durability. Understanding these layers and processes like tyloses formation helps us appreciate why white oak is such a valued material in woodworking and construction. Its natural resistance to moisture and decay makes it ideal for things like barrels, flooring, and furniture. So, recognizing these key features allows us to better select and utilize oak in our projects. Now, let's turn our attention to tyloses, a fascinating feature in wood anatomy. As highlighted by Butterfield, Malin, and Peslin, tyloses are outgrowths that develop from parenchyma cells into the vessel lumens. Essentially, they act as natural blockages within the vessels. These structures play a crucial role in the tree's defense mechanism, helping to compartmentalize and prevent the spread of decay or pests. Importantly, tyloses can also influence wood properties by affecting permeability and water transport. This means they can impact how the wood dries and its overall strength. Recognizing the presence and distribution of tyloses is vital for understanding wood quality and its potential uses. In the broader context of our presentation, tyloses exemplify how trees adapt structurally to environmental challenges. Their formation reflects an intricate balance between maintaining functionality and ensuring survival. So, understanding tyloses helps us appreciate the complexity of wood as a living, dynamic material.